Bifenthrin is a pyrethroid insecticide used primarily against the red imported fire ant by influencing its nervous system. It has a high toxicity to aquatic organisms. Although it is listed as a restricted-use chemical in the United States, it is allowed to be sold for daily use, provided the product sold has a low concentration of Bifenthrin. The chemical was discovered and developed by FMC Corporation. Products containing Bifenthrin include Talstar, Maxxthor, Capture, Brigade, Bifenthrine, Ortho Home Defense Max, Bifen IT, Bifen LP, Torant, Zipak, Scott's LawnPro Step 3, Wisdom TC Flowable, FMC 54800, and OMS 3024. Chemical properties: Bifenthrin is poorly soluble in water and often remains in soil. Its residual half-life in soil is between 7 days and 8 months, depending on the soil type, with a low mobility in most soil types. Bifenthrin has the longest known residual time in soil of insecticides currently on the market. It is a white, waxy solid with a faint sweet smell. It is chemically synthesized in various forms, including powder, granules and pellets. However, it is not naturally occurring. Like other pyrethroids, Bifenthrin is chiral. It has different enantiomers which can have completely different effects on the human body, which is a chiral environment. Bifenthrin is found in 2 enantiomers, 1S-Bifenthrin and 1S-Bifenthrin. 1S-Bifenthrin is 3 to 4 times more toxic to humans than 1S-Bifenthrin, while the latter is more than 300 times more effective as a pesticide. Toxicodynamics: the 2 types of pyrethroids are the ones with an I plus or minus cyano group and the ones without an I plus or minus cyano group. The neurotoxicity of Bifenthrin is based on the affinity with voltage-gated sodium channels. The pyrethroids with an I plus or minus cyano group block the closing of the sodium channel permanently, causing the membrane to be permanently depolarized. The resting potential will not be restored, and no further action potential can be generated. The pyrethroids without an I plus or minus cyano group, to which Bifenthrin belongs, are only able to bind to the sodium channel transiently. This will result in so-called after-potentials and eventual continuous firing of axons. The resting potential is not affected by these pyrethroids. Bifenthrin will open the sodium channel for a shorter period than other pyrethroids. The mechanism in mammals and invertebrates is not different, but the effect on mammals is much less due to higher body temperature, higher body volume, and lower affinity of Bifenthrin to sodium channels. Toxicokinetics: numerous studies have been conducted on the half-life of Bifenthrin in soil, water, and air under different conditions, such as aerobic or anaerobic, and at different temperatures and pH. It is more likely to remain in the soil and not so much in water, nor in the air. Because of the water insolubility of Bifenthrin, it will not rapidly cause contamination of groundwater. However, some contamination might occur by soil-bound Bifenthrin to surface water through runoff. For an overview of the environmental degradation of Bifenthrin, see figure below. The main path of degradation results in for a Euro unregistered trademark hydroxybifenthrin. Biotransformation: pyrethroids are much less toxic in mammals than they are in insects and fish, because mammals have the ability to rapidly break the ester bond in Bifenthrin and break the substance into its inactive acid and alcohol components. In humans and rats, Bifenthrin is degraded by the cytochrome P450 family. Use: on a large scale, Bifenthrin is often used against red imported fire ants. It is also effective against aphids, worms, ants, gnats, moths, beetles, grasshoppers, mites, midges, spiders, ticks, yellow jackets, maggots, thrips, caterpillars, flies, and fleas. It is mostly used in orchards, nurseries, and homes. In the agricultural sector, it is used in great amounts on certain crops, such as corn. About 70% of all hops and raspberries cultured in the United States are treated with Bifenthrin. Bifenthrin is not at all toxic to plants. Though it does not have a large toxicological risk towards mammals or birds, Bifenthrin is able to accumulate in food, so it might be dangerous to mammals or birds in some scenarios. Toxicology, toxicity in animals, mosquitoes. Bifenthrin is an effective pesticide to use against malaria and filaria vector mosquitoes. It is still effective when a resistance to other pyrethroids is found. Mosquito nets and indoor walls can be treated with Bifenthrin to keep more mosquitoes away. Bifenthrin is an effectively used insecticide, but the risk is high of it working only for a short time. Mosquitoes can develop a resistance to it, as well. Aquatic life. Bifenthrin is hardly soluble in water, so nearly all Bifenthrin will stay in the sediment, but it is very harmful for the aquatic life. Even in small concentrations, fish and other aquatic animals are affected by Bifenthrin. One of the reasons for the high sensitivity of fish is fish have a slow metabolism. Bifenthrin will stay longer in the system of the fish. Another reason for the high sensitivity of fish is the effect of Bifenthrin as AT-base inhibitor. The gills need ATP to control the osmotic balance of oxygen. If the fish is no longer capable of taking up oxygen because ATP can no longer be used, the fish will die. In cold water, Bifenthrin is even more dangerous. pH and calcium concentration are also factors that influence the toxicity. Vertebrates are less sensitive to the effects of Bifenthrin as AT-base inhibitor. Bees. In bees, the lethal concentration of Bifenthrin is about 17 mg per liter. At sublethal concentrations, Bifenthrin reduces the fecundity of bees, decreases the rate at which bee larvae develop into adults, and increases their immature periods. Table of LD50 values. Toxicity in mammals, chemical consequences. Bifenthrin and other synthetic pyrethroids are being used in agriculture in increasing amounts because of the high efficiency of these substances in killing insects, the low toxicity for mammals, and good biodegradability. However, because of his success, they are being used more often and high exposure of Bifenthrin to humans can occur. Cytotoxicity. Scientific tests in vitro have concluded that cytotoxic damage like DNA breakdown and gene mutation can be caused by Bifenthrin. Cytotoxic effects are not seen directly, but are more long-term. These effects can differ in severity based on to which enantiomer the body is exposed and the dose. Cytotoxic damage in humans results from increased oxidative stress, which leads to single-strand DNA breaking and degrading of lipids in the plasma membrane and F-actin. The exact mechanisms included in this path are not completely known yet, though several groups have found matching results regarding increased reactive oxygen species production which induces apoptosis. Bifenthrin also activates the LFA1/ICAM function of T cells. This function is a marker for inflammation. This means T cells are recognized by the body as if they were inflamed. This can ultimately lead to autoimmune diseases, such as asthma, bronchitis, and arthritis, and cancer. Neurotoxicity for humans. Exposure to Bifenthrin can be achieved either by skin contact or ingestion. Skin contact is not toxic, apart from a slight tingling sensation on the specific location of contact. Ingestion is toxic, although only slightly. Commercially available Bifenthrin, however, can induce toxic effects in those concentrations, because the added chemicals which improve the sustainability either potentiate Bifenthrin or are toxic of their own. Symptoms of excessive exposure are nausea, headaches, hypersensitivity for touch and sound, and irritation of the skin and the eyes. Regulation. The U.S. Environmental Protection Agency monitors and regulates the use of pesticides in the United States. Bifenthrin is classified as a restricted-use pesticide, meaning it is allowed to be legally sold only by certified pesticide applicators. For everyday use, it is allowed to be sold in lower concentrations. Bifenthrin has been approved for use against the raspberry crazy ant in the Houston, Texas, area, under a special crisis exemption from the Texas Department of Agriculture and the EPA. The chemical is only approved for use in Texas counties experiencing confirmed infestations of the newly imported, invasive ant species. The EPA has classified Bifenthrin as a Class C carcinogen, a possible human carcinogen based on a test with mice, which showed increased development of certain tumors. An acute and chronic reference dose for Bifenthrin has been established, based on animal studies. The reference dose resembles the estimated quantity of a chemical which a person could be exposed to every day without any appreciable risk of adverse health effects. The acute reference dose for Bifenthrin is 0.328 mg per kilogram body weight day. The chronic reference dose for Bifenthrin is 0.013 mg per kilogram body weight day. Bifenthrin was included in a biocide ban proposed by Swedish Chemicals Agency, because of its carcinogenic effect. This was approved by the European Parliament in 2009. Pesticides containing Bifenthrin were withdrawn from use in the European Union. They have since been reinstated. References. External links, pins and pyrethroids fact sheet, National Pesticide Information Center. Pesticide Properties Database record for Bifenthrin.